Hi, my name's Will Krawczyk, Scientific Services Manager at Chemical Computing Group. Today I'd like to show how to predict the structure of a protein-protein complex, including how to cluster docked poses, identify interacting residues at the protein-protein interface, and understand the specific details of these interactions. This is useful for identifying patentable, druggable sites or for selectivity analysis. So let's get started. Here I'm starting with a pre-prepared structure. The structure's been prepared using quick prep with the fix option toggled off. I've also annotated the antibody, which not only renders the structure in the main Mo window, but it also creates sets or collections of atoms that we can reference later when we set up the docking run. Finally, I switched the system manager from basic view to extended so that I can get control over each chain that we have in our system, so light, the heavy, and the antigen. In today's session, we'll be performing a self-docking exercise where we will redock the antibody to the antigen. Although this example is an antibody-antigen complex, the same approach works for docking any two proteins, or proteins to peptides, or even peptides to RNA. So I'll just go ahead and launch the protein-protein dock panel. This is a unified docking application whereby the methodologies are displayed on the left-hand side. The first four relate to docking of small molecules, and the final one allows us to leverage external programs. However, today we're using protein-protein docking, and regardless of the methodology that we use, the panel stays pretty much the same on the right-hand side. The top allows us to specify the receptor, middle for the ligand, and bottom handles refinement and scoring. Here Mo's done the heavy lifting for us, auto-detecting that the antigen will be used as the receptor, the one that's fixed in space. The antibody will then be used as the ligand, the one that moves around. So I'll just accept the defaults and initiate the calculation by clicking Run. A database viewer is opened where the results will be written. I just want to take a moment to look at some of the other options that we have in the dock panel. If we had information regarding the epitopes from the antigen, I'd be able to specify them here under the site. In today's example, I didn't know what the epitopes were, so for that reason, I leave the default to be all atoms. Likewise, if I know information on the paratopes, I can specify them here under the ligand site. By default, we'll dock to H3, which proved to be the most successful restraint during the development and benchmarking of this application. The Use Hydrophobic Patch Potential option is toggled on by default, and that's because we find that during development the best poses have a hydrophobic patch buried at the interface. So having this option on favourably weights those poses that have such a buried hydrophobic patch. We can also modify the number of poses that we'll be using, and you'll see here this is a funnel approach, so we can focus on only the most promising poses for the latter, more computationally intensive phases of docking. Now, in the interest of time, I'm just going to cancel this dock run and leverage some pre-computed results, which appear in the database viewer, which is the third major window type in Mo. We use this for storing database entries, or in this case, database results. It has a look and feel like an Excel spreadsheet, but you can see that it actually contains our molecules. And this is also our launching point to the various tools that we have in Mo. So for example, we could calculate protein properties for all of the molecules in our system. Likewise, we could do the same for protein patches, or perhaps we want to jump into some high throughput biologics screening. In this case, the database viewer holds the top 20 poses from our dock run, and you'll see that we've got fields for the molecule, which hold the antibody, receptor, which hold the antigen, and these are all ranked according to their score under the S field, whereby the more negative this value becomes, the better the interaction. If I just scroll along, you'll see that we also have other useful information, such as the protein-ligand interaction fingerprints, which we'll come back to in a moment. So to browse through some of these docked poses, I'll click File and Browse, which opens the Browse panel. I'm just going to make some changes to how we display our docked poses, so I'll render these in green and I'll just hide the atoms. We don't have a ligand present, so I'll just toggle off this 2D option, and I'll also write the score to the Browse panel so that I can keep track of that as I browse through some of these poses. As I click through, you'll see that the first few have good correlation to the original co-crystal, and as we go further down, the score drops off and we see some variation. So that's how we perform protein-protein docking. Next, we'll use epitope mapping for the goal of identifying interacting residues at the protein-protein interface. This is useful for identifying patentable druggable sites or for selectivity analysis. So I'll just go and launch this from the PLIF panel, and we see the epitope application appears at the bottom of the database viewer. So epitope predictions are made using the PLIF field from the database viewer that we saw earlier, and we do this for each of the docked poses. These are then clustered, allowing for an ensemble analysis of our docked poses. Epitopes are then identified, ranked, and saved. So here we can see that we've got 5 clusters arranged by score. Cluster 1 has 6 poses associated with it, and seeing lots of poses in the same cluster provides higher confidence that these epitopes might be involved in binding. The remaining clusters have 1 pose per cluster. The bottom of the panel allows for visualizing the epitopes and their associated poses, plus options to further analyze epitopes in other Mo applications such as PLIF, which we'll do in just a moment. So I'll just go ahead and select that first cluster, and I'll select the entries in the database. And when I do that, you'll see that 4 out of the top 5 poses are associated with that first cluster. If I toggle on the Annotate button, that will allow us to visualize the epitope residues that correspond to cluster 1. If I just hide the original co-crystal, it makes it a little easier to see. So you can see here on the antigen ribbon, in yellow, our epitopes are displayed. I could also toggle on the Show Poses so that we can have a look at the variation of the docked poses that are associated with that first cluster. So from here I could click through and have a look at some of the other epitopes associated with some of the other clusters. I'm just going to toggle off the show poses, and next, to better understand the frequency and the detail of the interactions associated with each epitope cluster, I'll leverage PLIF by clicking PLIF Analyze. So this opens the PLIF panel, which has a barcode representation of the interactions at the top. So the top part of the panel is telling me whether an interaction occurs or not. The middle part of the panel displays the epitopes that are involved, and the lower portion provides detail on the type of interaction. So here I can see that we've got the 6 rows for the 6 poses associated with cluster 1. Reading from left to right for the first pose, I can see that we don't have a barcode present, so it's telling me there is no interaction with methionine 1. We do get a barcode on proline 3, so there is an interaction there, not on proline 6, and so forth. To understand the type of interaction, I'll refer to the bottom part of this panel, and I can also leverage bubble help here, which will give me information on the letters that represent the different types of interactions. So for example, C and H would be a total surface contact and a hydrophobic surface contact. So in this case, I can see that we've got 2 arginines and a glutamic acid that are involved in all 6 of the poses associated with that first cluster. And if I just scroll along looking for residues 108, 109 and 111, I can see that these have a variety of total surface contact and charged surface contact interactions. That brings us to the end of today's session, where we use protein-protein docking to predict the interaction between 2 proteins. We then identified epitope residues involved in binding and visualized them on the 3D structure. This is useful to identify druggable sites or for selectivity analysis. Finally, we use PLIF to identify the detail of these type of interactions. If you have any questions, please do reach out to us at support@chemcomp.com.